Our nervous system helps us understand our environment. It receives sensory information, and this information is received through various sense organs such as eyes, nose, ears, skin, and then this information has to be processed and interpreted. So this information is taken from the sense organs to the central nervous system, where this information is processed, and the central nervous system decides how to respond to that stimulus or that information, and then some motor responses are produced. So these responses are produced by activating some muscles or glands. This kind of mechanism of receiving information and producing some responses help in maintaining not only our interaction with our environment but also our internal stability or our homeostasis. How? By understanding changes in pH or temperature or some kind of fluid imbalance, we are able to make some adjustments in our internal processes to adjust or to cancel out those effects and maintain a homeostasis. So what carries the information from sense organ to the central nervous system, and from central nervous system to different effector organs? These are the specialized cells called neurons. And this is the diagram of a typical neuron. A neuron has a cell body where you find its nucleus and other important cell organelles like mitochondria, ribosomes, or endoplasmic reticulum. And there are several extensions coming out from the cell body of a neuron, which are basically the extensions of plasma membrane, and these extensions are called dendrites or axons. The difference between the two is the impulse is received from the dendrite side, or this is the signal receiving side of a neuron, and the signal is passed down the length of axon and it goes to the axon terminal. Here you will see that the length of axon is wrapped around by the extensions of another cell called the oligodendrocyte cells, and these extensions are wrapped around the axon. The function is to provide insulation to the axon and helps in faster transmission of impulse, because the impulse can jump from one node to another node to another node to another node. Now where the impulse reaches, this is called the axon terminal. It has to make a junction with the next neuron, and that junction is called synapse. Here neurotransmitters are released, and the action of those neurotransmitters will then onset the impulse or electrical conduction of signal in the next neuron, and we'll study about that later in this talk. There are three different types of neurons based on their function. The sensory neurons, which transmit the signal from the receptor organ or the sense organ to the central nervous system, they look like this. So we can call them a pseudo unipolar neuron, because from the cell body it seems like one extension is coming out, but then it bifurcates and one end ends up behaving like a dendrite side and the other end ends up working like an axon side. An interneuron is a neuron which is present in the central nervous system, and they are basically connections between sensory and motor neuron. So an interneuron is something like this, which is a bipolar neuron, one side extending from the cell body is dendrite side and the other side extending from the cell body is the axon side. A motor neuron is the one that takes the signal from the central nervous system to the effector organ or the muscle or gland that has to produce an effect in response to the information or stimulus that has been received by our body. And these are typically multipolar neurons, because from the cell body you see that there are many extensions coming out, which is why it's called a multipolar neuron. So here we learned that Neurons are basically the functional cells of nervous system. But other than that, there are different kinds of supporting cells present in the nervous system. These supporting cells are called neuroglia, and there are several different kinds of cells classified as neuroglia cells that serve different kinds of functions in the central nervous system. For example, astrocytes create a blood-brain barrier between the central nervous system and the circulating blood, so that the stuff that is in the blood, for example, there can be microorganisms, pathogens, or some metabolites that you don't want to pass through the central nervous system, that that barrier is created by astrocytes. Then there are ependymal cells. These cells secrete cerebrospinal fluid, and cerebrospinal fluid is the circulating fluid inside the central nervous system. Oligodendrocytes that we saw before are cells that make these extensions that wrap around the axons, and they not only provide insulation but also speed up the conduction because the nerve impulse starts jumping from node to node. Microglia cells are immunological in function. They remove the dead cells and pathogens from the central nervous system. Then we have satellite cells. These cells regulate the neurotransmitter levels, and any changes in neurotransmitter level are related to so many psychological issues which people face. For example, depression, anxiety, and hallucination. So these symptoms are regulated by the neurotransmitters. And the last cell we are going to talk about are Schwann cells, and these cells also create myelin sheath, but we'll learn what is the difference between central nervous system and the peripheral nervous system. So oligodendrocytes make myelin sheath in central nervous system, and Schwann cells make myelin sheath in the neurons of peripheral nervous system. So now we'll study how a neural signal is transmitted. So everything starts with receiving a stimulus. Any change or any information in the form of touch, light, or chemicals are detected by sense organs. And our sense organs are eyes, tongue, nose, ear, and our skin. So receiving that stimulus causes some change in the potential of the cells or neurons present at the site of those sense organs, or the receiving point of that information. But what does that mean? What does it mean that the membrane potential is changing? So imagine that this is an axon or a neuron. So neuron has a high concentration of potassium ion inside and high concentration of sodium ion outside. This high concentration of sodium ion outside and potassium ion inside is constantly maintained by spending a lot of energy in sodium potassium pump. So there is a pump in the membrane of these neurons which are constantly working, that throw sodium ions out and bring potassium ions in, and this effort of this pump requires energy. So that's why we call it an energy expensive process, or this system is called pumping, because we are using basically energy to pump a system that throws sodium ion out and potassium ion in. So because of sodium ion concentration being higher outside and potassium being higher inside, there is a potential difference across this membrane, and this potential difference is about minus 55 to minus 70 millivolt. So we consider that inside of the membrane is negative when there is no impulse in the neuron, or neuron is at rest. So that resting potential is shown here, and resting potential is about minus 70 to minus 55 millivolts. So when an impulse is received or a stimulus is received, it causes changes in the local membrane potential. That means this potential starts to change. So what basically changes is the sodium ion flows transiently for a very short amount of time. Sodium ions flow inside the neuron. This makes the inside of the membrane less negative. So you will see that there will be certain increase in the positivity or decrease in negativity of the internal environment of a neuron. But this sodium ion concentration that comes in and tries to make the potential less negative should be high enough. That means it should at least be up till the threshold level. Only then a neuron will fire an action potential, or a neuron will feel that the stimulus is strong enough and now I have to send this information to the central nervous system. So that time it's the impulse is triggered, and that impulse will be traveling to the central nervous system. If the stimulus is not strong enough and it has not crossed the threshold level, the axon will not worry about sending that information to the central nervous system. So as mentioned earlier, at rest there is more potassium inside and more sodium outside, leading to a net more positive charge outside and comparatively negative charge inside. So there is a negative potential difference between not negative potential difference, but a negative potential inside, and there is a potential difference of about 70 millivolt between the inside and outside of the membrane. So this is called the resting potential when the neuron is at rest. Now let's say a stimulus is received and the inside environment becomes less negative. So as you can see along this axis, we are becoming less negative. That means the sodium starts to flow inside the cell, making the internal environment less negative. Now let's say this stimulus is strong enough and it has crossed the threshold. Then what happens? Sodium channels will open. That will lead to a rush of sodium entering into the cell, and the inside of cell starts becoming more and more positive, more and more positive. So at this stage we had a potential which was very strong, about minus 70, minus 75, minus 80 millivolt, but now it is slowly reducing and almost reaches to 0. That means inside and outside ion concentration becomes almost similar. But then it overshoots such that you have more sodium inside and less sodium outside. So much positively charged ion has started coming in with all the potassium and sodium being inside. It makes the internal environment very positive. So you can see now potassium is there and sodium is there, so internal environment becomes very positive. Now at the point of repolarization, the potassium channel opens, and the opening of potassium channel will start to throw potassium out. That means slowly the positive charge of inside of the membrane starts to decrease. Now positive charge will start decreasing and it will become more and more negative. Slowly it becomes more and more negative and almost reaches the level where it started. It becomes more and more negative. But the problem is, by the end of it, there will be more sodium inside and more potassium outside, which is reverse of what we had in the beginning, where we had more potassium inside and more sodium outside. Okay, so now the potassium that's going out doesn't really stop there, but overshoots this baseline and becomes hyperpolarized. So a lot of potassium ends up going outside and compared to the amount of sodium that we have inside. Now we need to bring this back to the baseline level. How will we do that? Again, the sodium potassium pump will start to work again. That will throw sodium outside and bring potassium inside, and we will revert back to the situation that we started off with. So we have learned what is happening, how internal environment was negative in the beginning, and then for a short amount of time becomes positive, and then end up reverting to its original situation. But why are we doing that? So here you will see that this small area becomes positive for a short amount of time, and you see that there is depolarization. It's becoming positive, more and more positive inside. And then you see that this area becomes uh, back to the original situation, inside negative, outside positive. So it has reverted back to its original position. What is happening in the next area? Now next area is becoming positive, and the previous area has returned to its original situation. Now look at the area B. Now area B is inside negative again, and the next area is becoming positive. So slowly you will see that this positive charge is progressing forward, with the previous area reverting to its original situation. So it was positive here, then it became positive here, then it became positive here, this got back to original. Now it became positive here, this got back to original, then this became positive here, and this got back to original. So slowly we are progressing the positive charge along the length of the axon. But why are we doing this? So we are taking this positive charge down the axon, and it has to reach the axon terminal. When this positive charge reaches the axon terminal, what happens is the next step of conduction of nerve impulse. But one thing important to note here is, if this charge change has to happen along the length of axon, it ends up becoming a very long length to cover. But because of the myelin sheath present here, that covers most of the axon, this positive negative charge can only jump from one node to another, and this conduction becomes very fast, and this kind of conduction is called the saltatory conduction. So we learned that when an impulse is traveling down the length of an axon, it's trying to actually send the positive charge at the axon terminal. This positive charge that reaches the axon terminal is then able to open the calcium channel. So the goal was to open these calcium channels. Then what happens? These calcium ions rush into the axon terminal. These ions trigger these vesicles to move to the plasma membrane and perform exocytosis. So these vesicles contain neurotransmitters, and when these vesicles fuse with the plasma membrane, the neurotransmitters are released in this space between the two neurons. So this is the neuron that's bringing the impulse, and this is the neuron that will receive the impulse. So the electrical impulse reaches the synapse, that leads to the calcium ion going in, which triggers the release of neurotransmitters. Then these neurotransmitters bind on the receptors of the next neuron. So these neurotransmitters can be acetylcholine or dopamine or serotonin, and on the next neuron they are supposed to be receptors which can receive dopamine signal or serotonin signal. So for every neurotransmitter there are specific receptors present on the next neuron. So this next neuron is called the postsynaptic neuron because its presence is after the synapse. So once the neurotransmitter sits on the receptor, it can either excite the next neuron or inhibit the next neuron depending on the nature of the neurotransmitter. So you can increase the stimulation of your body or your nervous system, or decrease the stimulus of your nervous system, both depending on your physiological conditions, and that helps in maintaining the working or functioning of your nervous system. Now let's look at the divisions of nervous system. Your nervous system is divided into two main parts. The central nervous system, which in this picture is indicated in pink color, it contains brain and the spinal cord. And peripheral nervous system, which contains all these yellow colored nerves that are arising from brain as well as spinal cord. So the nerves that arise from brain are called the cranial nerves, and those arising from the spinal cord are called the spinal nerves. So the central nervous system mainly processes and integrates information, and the peripheral nervous system is associated with relaying that information. It gathers information from all the sense organs, brings it to the central nervous system, and also take the response of information from central nervous system and takes it to the affected organ. Brain is the main organ of human beings that makes them highly evolved. It has three main parts. First is the forebrain. Forebrain is associated with higher thinking, sensory responses, voluntary actions, emotions, and memory. So the forebrain has these main parts. The first, the big brain that is visible from outside and has all these folds, is called the cerebrum. It's mainly associated with thinking, learning, and voluntary movements. If you open or separate the two hemispheres of brain, then the part of brain which is hidden inside will be visible. In the center we have thalamus, and thalamus is associated with relaying the information to different parts of the brain. At the base of thalamus, you will find hypothalamus. This is the area which is below thalamus, which is why it is called hypothalamus, and it regulates hunger, thirst, body temperature, emotions, and also talks to the pituitary gland and regulates its functions. Another part of our forebrain is the limbic system, the system that helps us with emotions and memory, and two important parts of limbic system are hippocampus and amygdala. The next part of the brain is the midbrain. Its main function is to deal with the reflexes, motor responses, and process the visual and auditory information. This part of brain is called the midbrain, and the midbrain has some regions of neurons called corpora quadrigemina. They contain superior and inferior colliculi, which process visual and auditory signals respectively. Other than that, you have stalks of cerebral peduncles which connect forebrain with the hindbrain, and they transmit information about the motor signals. A part of midbrain which is not visible in this picture is called substantia nigra, and substantia nigra has neurons which release dopamine, and as we have heard about dopamine so frequently, that it is associated with our reward mechanism. It helps us feel accomplished when we perform a task, and if that neuron is affected, or the neurons that produce dopamine are affected, it leads to Parkinson's disease. The last region of brain is called hindbrain, and hindbrain is involved with the regulation of basic life functions such as breathing, control of heartbeat, coordination of body posture, and balance. And it has three main parts which are visible here: cerebellum, pons, and medulla. Cerebellum is involved in the regulation of voluntary movement, posture, and balance. Pons connects the cerebrum with cerebellum and regulates breathing, and medulla oblongata is again involved in the regulation of life functions such as heart rate, breathing, blood pressure, and reflexes. The second part of central nervous system is the spinal cord. Spinal cord starts from the medulla and then extends down in the vertebral column. So it extends up till first or second lumbar vertebrae. It's protected by the vertebral column or the vertebrae and the linings on top of it, which are called the meninges, and inside it is the cerebrospinal fluid. The spinal cord has 31 segments, and therefore 31 pair of spinal nerves arise from it. And spinal cord can also be divided into 5 different sections, just like your vertebral column. Structurally, inside of the spinal cord, which you can see in this picture, kind of looks darker and the outside looks lighter. So inner part is containing the cell bodies of neurons, and it's called the gray matter, and the outside is contains the axons, which is why it appears lightish in color because of the presence of myelin sheath around them. The function is the gray matter processes the sensory and motor information, and the white matter is basically associated with conduction, just like the axons are associated with conducting information from one place to another. Overall, the spinal cord carries information to and from the brain to different parts of the body and controls the reflexes. So reflexes are those actions about which we don't think much. They are very immediate responses. For example, shutting your eyes when light flashes in your eye, or pushing the brake when you suddenly feel that somebody is coming in front of your car. Those reflexes have to be very sudden and very fast. So we don't want to spend time in sending that information to the brain, thinking about it, and then performing the function. But we bypass all that and take shortcuts to respond to those situations as a survival mechanism, because if we don't react very fast, our chances of survival in those situations will reduce. Now that we have talked about the central nervous system, let's talk about peripheral nervous system now. Peripheral nervous system is composed of all the nerves that arise from the brain and spinal cord and spread all over the body. So the peripheral nervous system has 2 branches depending on how we respond to some information. Are we responding in a more voluntary method by thinking about it, by processing that information and under our own will, or we are responding to the information in a more autonomic or involuntary method? So the involuntary responses that we don't really think about, but our body keeps on doing them themselves, are of 2 types: sympathetic and parasympathetic. So sympathetic responses are those that we perform when we are in a stressful situation, when we are preparing our body for fight and flight responses. And parasympathetic responses are those which we perform at the time of rest and when we are not really in an emergency situation. If we have to think in terms of the bodily functions, in parasympathetic situation, when we are more in a resting phase, our pupils are constricted, we are salivating because we are enjoying our food, our heartbeat is pretty normal, the ventilation in lungs is also calm, and we are more focused on enjoying food, digesting. That's why our digestive system or gallbladder, intestine, and our excretory systems are more active. Whereas when we are in a panic situation, we want to focus more on the information, and our heartbeat is racing. We want to send oxygen and nutrients to all the parts of the body because we want to produce more energy and be ready for fight or flight responses, and we are hyperventilating. At that time, we are not really concerned with the digestion process or excretory process. That's why the functions of alimentary canal are kind of reduced, and salivation is also reduced because our resting, digesting, and enjoying phase is kind of suppressed, and our fight and flight responses are more active in sympathetic responses. There is a vast array of nervous system disorders, but some of the main ones are Alzheimer's disease, which is associated with loss of memories due to degradation of our neurons. As you can see, the shrinking of brain is seen in Alzheimer's patients. Parkinson's disease is associated with loss of neurons that produce dopamine, so body not only loses its pleasure and response system, but also the coordination of our voluntary movements. Epilepsy is the situation of frequent seizures in which the neurons are firing continuously in a very uncontrolled and uncoordinated manner. Multiple sclerosis, or also called MS, is an immunological disorder in which the myelin sheath is being destroyed or attacked, and that leads to the loss of insulation over the neurons. Stroke is the condition where the blood vessels in our brain are blocked, and that leads to reduced functioning of brain because the brain is not getting enough nutrients and oxygen to perform its work. To maintain a healthy nervous system, one should regularly exercise to promote blood circulation to brain. They should eat a balanced diet which has omega-3 fatty acids. Enough sleep is important for consolidation of memory and cognition ability. One should try to reduce stress. That will reduce the cortisol level and help in managing your sympathetic nervous system. Avoid neurotoxins such as alcohol, drugs, and smoking. So today we learned about the nervous system, what are the components or main cells of nervous system, and how an impulse is transmitted. What are the parts of a central nervous system, peripheral nervous system, and we also learned how our body deals with sympathetic and parasympathetic responses, and what are common disorders of nervous system.